And this is going to be a very broad and general talk because we are partway through our summer project. We don't have a ton of data to show yet, but we're on it. Camilla and Matt are here with me, and we've had a relatively good last few days. And so we're hoping that by the end of the summer, we'll have a preliminary analysis of some of the killifish data for New Bedford Harbor, which will really be our focus. All right, so sorry, I'm not used to this Zoom thing. All right, I'm lying. I'm just bad at it. So here's the plan for my talk. I'm going to talk a little bit about rapid evolutionary change and how really common it is. I'm going to talk about some environmental successes. I think a lot of the time we feel paralyzed by how bad the news about the environment is, but we've solved a lot of really challenging problems. We don't talk about them much because they're no longer problems. That actually gives me great hope. Still, there are places that are a mess, like New Bedford Harbor. Global warming is, of course, a big concern. We'll talk about both those things. And then the heart of the research part of the talk is going to be talking about the Atlantic killifish. I'll talk a little bit about the biochemistry of how it detoxifies organic pollutants. We'll talk about our project plan, the technology we're using. I'll show some preliminary proof of concept type results. I'll do that pretty quickly because it really is just pilot data. Everything's working, but the three of us have a lot to do to make this work by the end of the summer, but we are very much on our way. And we are on our way thanks to all the little supporting funds that we've been able to put together. We're able to get salaries from the SURF program, of course. That's what most of you are supported by here. That is wonderful. That lets us have Matt and Camilla and my other former research students around all summer. That is vital. We have some support from the SNEC Foundation, who have been very helpful to PC. PC is also really good at supporting students to do research. We've got small research grants for students. Faculty are pretty well supported with small piles of competitive money that we can use to get projects off the ground. By putting these all together, I think we've been able to put together a pretty neat research program. All right, so Jim, a couple days ago, asked me whether I wanted to keep the same draft title I sent a month or so ago. And I said, yeah, and I started to think about my title. When I was in college, the dominant model of evolution was that it was a long, slow process. That's what Darwin said, small changes over time. But even then, we knew of a lot of contradictory examples. Agriculture is all about rapid evolutionary change. Here's a classic example that was taught to me as sort of exceptional back when I was a college student. The peppered moth, industrial melanism, as more coal was burned in English cities, the branches of trees got darker and darker, and the dark morph of this moth, which you can sort of see here and very well there, became more and more common in urban areas, and it hadn't been seen before the 1800s. Here's a lighter morph of the moth. Can you see my arrow? He's down in here. You can barely see that moth. And the idea is that if visual predators are around, if you're in a place with lots of soot-covered branches, you're going to be better off to look like this guy than that guy. Cool. That's a neat story, and that's been studied continuously since then. So one of the things I learned while I was getting ready for this talk is people believe now that changed, this new morph that really wasn't well known before the 1800s, was a result of a transposable element inserting into the biochemical cascade that controls the color. And that's how the dark colored morph evolved so quickly. And then natural selection made it more common in areas where the tree branches were dark due to coal pollution. Um, I'm bringing this up because this is kind of a neat coincidence. So this is Matt Rock. He is a rising junior here at PC. He's working with us on the Fungulus adaptation project, but he's also got a project that he got funded through PC to look for how transposable elements might be causing rapid evolution in fish. We're getting some cool, interesting preliminary results. I'm looking forward to putting that story together with him over the next year or so. It's going to be a neat story. It's kind of a lot. I'm appreciative of the fact that Matt is willing to change computers over so they run on Linux or whatever and make that part of the bioinformatics work. I can only help him so much with that, but I'm excited and I'm tickled to death by this coincidence. You know, transposable elements were responsible for the rapid evolution in this classic evolutionary system. Oh yeah, now we can hunt for those in different groups of fish that we have access to. Another classic example, this was long before I knew the grants, in the 1970s. There was a drought in the Galapagos Islands. The grants measured and banded all the birds every year for 40 years, I think the story is. It's a pretty amazing story. In the mid-1970s, there was a drought. That decreased the number of seeds, and that meant that you had to eat bigger seeds in addition to the littler seeds you preferred. Well, here's the distribution of beak depths before the drought. Here's how they are in the population after the drought. So all they did is measure from here to here, cataloged all that data, observed the population for a few years, and they saw that beak depth changed by almost a millimeter from here to here as a result of that drought. So that's real heritable evolutionary change. It has a genetic basis. So another classic story. I want to introduce you really quickly to one of the weirdest places I've ever been. This is a picture of the Salton Sea. Do you know about this, Jim? You're smiling. It is a cool place.
Hey Jeff, real quick, can I just say something real quick?
Yep.
I can't see your cursor when if you're pointing at things. I don't know if anybody else can, but
nobody's seeing it. All right. I don't know. Oh, can you see it now?
Yes.
Okay. So I need to do it on the other window. Thank you for that, Jim.
Okay.
All right. All right. So this is the Salton Sea, this dark area. It's east of San Diego. It's inland. I like to say it's one of the biggest lakes that nobody has ever heard of. It's, you know, it's the size of our state almost, or at least within spitting distance. And it's a weird place. It's an accidental lake. So, and it's a product of human intervention. So in the early 1900s, there was a big water diversion project diverting parts of the Colorado River for irrigation. Something broke and this low-lying basin got filled with water, and that became the modern Salton Sea. Now, because this lake is below sea level, there's no outflow. So the only way for water to leave this basin is through evaporation. And there's a lot of it because it is out in the middle of the desert. So this lake has been getting saltier and saltier and saltier over the last 100 years. In the 1950s, it was sort of like current seawater. These days, it's well above 50 parts per thousand. This is a cool evolutionary story. So one of the things they tried to do in the 1950s is turn this lake into an inland resort. I don't know, it's not too far from Palm Springs. They tried to recruit like Frank Sinatra and Dean Martin and those kind of Rat Pack guys to hang out there. They did for a bit. And the idea was they were going to turn it into a resort. As part of that, what they did is they introduced marine fish. So the idea was, hey, We can have a sport fishery here and people can come and water ski in the desert and they can catch fish. That'll be a blast. Well, the marine fish went extinct. They couldn't tolerate the ever increasing salinity. On the other hand, tilapia got in the lake. Tilapia are not native to California, of course. They're an African fish, but they were being farmed on a nearby fish farm. They got into the lake. They are a freshwater slash brackish fish. And over the last 50 years, they've managed to survive in this ever increasingly salty environment with levels of salinity higher than they ever experienced before. And I think that's going to be a pretty cool short-term evolutionary story. All right. So just to show you, so the idea to develop it as a tourist site didn't work out. It's kind of a modern day ghost town if you ever go there. This stuff on the rocks is salt and some barnacles, and then there's just lots of abandoned 1950s and 1960s optimism there. All right, so to transition a bit, I just want to leave you with the idea that rapid evolution is no longer thought of as exceptional. There are just tons of examples now. Evolution is a vital, ongoing process that we can observe in real time during our lifetimes. All right, now, Jim mentioned my other interest, and these things I hope are linked. The environment is always changing, and I'm really interested in studying genetic diversity. The more genetic diversity a population has, the better chance it has to have the right combination of alleles and therefore phenotypes to survive in a changing environment. This next slide, this is a quote I found a few years ago when I was writing a paper with my friend TJ McGreevy. This was written by Congress people in 1973, and it kind of makes me cry. This is really beautiful. In 1973, politicians recognized that genetic reverse diversity was a vital resource for our future ongoing human health. These are amazing resources we need to maintain genetic diversity in populations. And politicians understood the science well enough in the 1970s and were willing to be passionate about it. Wow, that's so cool. Also makes me slightly sad today. All right. So I promised you we would talk a little bit about things that give me hope about the environment, and then we'll talk about some of the challenges and relate that back to fish. Sometimes I think when we get sad and frustrated by how bad the environment seems to be and these intractable problems, we forget how much better we've been able to make things in my lifetime, if not yours. So this is water quality in the Connecticut River. The first time point was 1969, and now here we are in 2019. Phosphorus contamination. Phosphorus is, of course, part of many fertilizers. The runoff goes into rivers and streams, causes algal blooms. They die, they suck up all the oxygen, and terrible things happen. All right, so phosphorus out of place is pretty bad. It's a limiting element, ideally. Levels at this one station on the border between Connecticut and Massachusetts are down 74%. That's got to be a huge success. There are lots of examples like this. This just happens to be one that I found easily Googling. Air quality. Even more recently, 1980. By 1980, I was driving around in cars, and back then, there was still a lot of lead in gasoline. You could still buy leaded gasoline. We decided to change that. Lead levels in air are down 98% since then. That's huge. Sulfur dioxide. Jim mentioned that I was a grad student at the University of Vermont. This was in the early 1990s. Back then, when you go hiking, you go to a mountaintop, you'd see a lot of the pine trees were brown. That was thought to be due to the effects of acid rain, sulfur dioxide turning into acids in the atmosphere coming from power plants in the Midwest, blowing east. eroding the treetops in places like New England. We don't talk about acid rain very much anymore. We solved it. The levels of sulfur dioxide, which causes the acid, are way down. Nothing terrible happened in our solving it. So we've had huge successes over the last 20 or 30 years. I take a lot of hope from that. But there are still some real messes that we need to clean up. I'm going to transition now and start talking about, well, first of all, I'll just reiterate how amazing this is, despite the fact that the population between 1970 and now has climbed a lot. Well, those successes are really impressive. You know, if we carried on the same path, we'd be all suffocated by now and would have no water to drink or anything. All right. Now, there are many challenges. One of these are these persistent pollutants, and that's the story in New Bedford Harbor. So this is New Bedford, south of us here. It's about, I don't know, 40 minutes away from us. Here's its history. So in the early 1900s, there were a few electronics manufacturing plants. They discharged into the bay heavy metals and an organic pollutant called PCBs. This was all made illegal, and all this stopped by the 1970s. In 1982, the upper part of New Bedford Harbor was declared to be a Superfund site, liberating federal resources for cleanup. We're not done cleaning up yet. That is a long-term, ongoing project. The remediation solution is mostly dredging the sediments. They're removing the sediments from the bottom of the harbor, storing it someplace safe. That's another story. In 2020, the main dredging efforts finished, although they are still working around the edges. The kind of contaminants that are present are things like copper, chromium, mercury, cadmium for heavy metals. And then the organic pollutants primarily are PCBs, although there is a mix of other things. They came primarily from this kind of factory. This is the Aerovox plant. Again, it was closed in the late 1970s. I find it ironic that one of their slogan was products that endure, since we are still cleaning up 40, 50 years later. All right, just a few, this blows me away, just a few scary numbers. So in the late 70s, these folks measured the sum of the heavy metals, and they found out they made up to 1% of the sediment. That's high enough that these folks suggested that one way to remediate this problem is to sell mining rights. You know, pay people to dredge it up and take the copper out, and everyone will be rich and happy. That didn't happen. I don't know why. I suspect it's because the other contaminants in there make it too hot to handle. Another thing, so PCBs are pretty bad for us. I'll tell you why in a few minutes, but they make up, in some parts of this, in hotspots, up to 10% of the dry weight of the sediment is some kind of oily organic pollutant. That also seems like a lot to me. Problem is, these things don't biodegrade, right? They can get passed up the food chain, they bioaccumulate. Top predators die, a lot of them just fall back into the sediment, and the cycle repeats, cycle after cycle after cycle, in this case for decades. The human health effects of this are pretty well known. Heavy metals do all these bad things. PCBs are also carcinogens. They can do liver damage. They're endocrine disruptors, and that can mess with, among other things, our thyroid hormones, which regulate our metabolism, in addition to causing reproductive and developmental problems. Here's a map of all the dredging they've done. So the green is places where they've removed the harmful sediments. The pink are these intertidal areas. They are still working on this. Just to give you a sense of scale and place, this is 195 for those of you who may have driven through. So this is the width of a 4 or 8 lane highway at this point. A pretty big area. They're also dredging down here. This is a hurricane barrier, which fortunately seems to be holding many of these pollutants in. I was talking to Matt the other day, and he asked me a little bit about human health effects, and I did some digging. I was surprised, and again, I'm new to the system, so this knowledge may not be complete, but I wasn't able to find any epidemiological studies that showed what impact this might be having on humans. But when you go there, it smells oily. It's like when you go to the basement of someone's house where they have oil heat, or when somebody's pumping diesel into a car or a trailer truck, it smells like that, despite all the dredging and remediation that's been done. This paper suggests that at these levels, there will be human health effects. It's interesting, it's theoretical. They've measured the levels in the air, and this is what you see if you live a couple meters away from the shore. They, as far as I could tell, haven't identified any disease clusters, and certainly breathing less pollution is better than breathing a lot of pollution. So it's an interesting and kind of scary environment. I'll go through this really quickly. These are just levels of pollution in the different areas of this bay. As you can see, most of the pollution was up here. Now, this is over the course of dredging. These numbers represent the average level within a segment, and this is parts per million of PCB. And so early on, it looked like the levels went up. I assume that's because they were disturbing some of these sediments. Now, as of 2009, levels were much lower. I mean, they're putting it in green, but man, that is still a lot higher than it is outside in these cleaner areas. So there's still a lot of stuff there. Now, this is 10-year-old data. I'm not sure what's going on today exactly in terms of the numbers. How does this affect the biological community? Doesn't do it any good. So what we're looking at is an index of biodiversity. This is basically a variant of something like Shannon's index, where you're looking at a lot of creatures in a defined area of settlement, and you're counting up the number of individuals and number of species. So a more diverse, more healthy community has a high index value, and it's very low when the community is heavily impacted. This is dominated by a small number of species. This is lots of different creatures. And again, this is over time, starting in 1993. As you can see, the most impacted parts are getting better, but man, they are only 20% of the level of the worst parts. One of the creatures that has managed to thrive in this environment is the Atlantic killifish. This is an amazing evolutionary story. So people noticed that Atlantic killifish, Fungulus heteroclitus, also mummichog, I'm going to alternate between the three. Some of them survived in these very polluted environments. A sad part of the story is New Bedford Harbor isn't really a one-off. There are lots of polluted estuaries up and down the East Coast. People noticed that, hey, Fungulus is doing well in New Bedford Harbor. It's doing well in polluted parts of Connecticut. It's doing well in polluted areas in New York, and it's doing well in polluted estuaries in Virginia. They have evolved to tolerate these high levels of PCBs, and it's pretty amazing what levels they can tolerate. We know this is a real evolutionary story. People have captured fish from polluted environments, and they've captured fish from clean environments. They've raised them in the lab for a few generations. When you expose fish from whose ancestors came from clean environments to PCBs, they die. When you expose fish whose ancestors came from polluted environments, they do okay. And what's amazing to me is they can survive levels of PCB that are 8,000 times higher than the level that will kill fish whose ancestors came from clean environments. So it's an example of rapid heritable evolutionary change in the last 40, 50, 70 years. A pretty cool story. Now, this is not really Jeff's story. I really want to thank Diane Nachi, who was my postdoc advisor at EPA in the early 2000s. She's the one who really pulled this story together. Um, I'm also working with Dr. Brian Clark, who's also at the EPA. He has a long history of working on this system as well. He's worked out some of the biochemistry. He's written a couple awesome review papers that I use in teaching. Um, and they've really pulled the story, big parts of the story together, in addition to some folks at Woods Hole and a lot of other researchers. I was very flattered when they invited me to come in and start to explore the population genetics of this system. All right, so we know a little bit about how these adapted fish manage to do this. This is, I'm sort of getting outside my comfort zone, but what is a summer surf for except to get outside your comfort zone? So this is a biochemical pathway that many vertebrates use to detoxify many different organic toxins. I don't understand the whole enzyme cascade, but what I do understand is people have sequenced these and looked at variation across tolerant and susceptible populations and found there are differences in several of these different genes involved in this pathway. What amazes me, and this seems so counterintuitive, fish who have adapted to survive in this bad environment do not have a super amazing version of the system. Instead, they just give up. They turn the whole system off. So the mutations that fish who live in these polluted environments have are ruining these proteins and they don't work anymore. I don't understand that very well. I've talked a lot to Brian and Diane. They understand it quite a bit better. The take home I get from them is, well, if you're exposed to run-of-the-mill organic toxins, this is a great system to have. If you're exposed to PCBs and these artificial toxins, the breakdown products from that down in the enzyme cascade are actually just as bad or worse as the PCBs. You might as well not bother. I think the other part of the story is that the other part of the story is that running a system like this probably takes a lot of energy. You're using a lot of energy to detoxify things. If that's making you exhausted, maybe you're better off not doing that. Now, Brian is very interested in understanding the trade-offs for that. You know, if you can survive without the system working, why do you bother to have it? That's going to be a more complicated story, and I look forward to seeing how that develops over the next few years. So far, there's no smoking gun that says, hey, if you turn this thing off, something bad happens to you. But still, we know it must be true. And this is a very conserved system. We all have it. Most vertebrates have it. All right, so here are our general study questions, and I do promise I'm sort of in the home stretch. I'm interested in understanding how much migration occurs between estuaries. Lots of migration means lots of genetic connectivity. Big picture, to bring this back to the idea of genetic diversity. If you're part of a big connected population, that means you have all kinds of genetic resources to make different phenotypes to adapt to novel pollutants like PCBs. On the other hand, if your populations are very isolated, your population got really lucky if you're surviving some new environmental challenge. I'm curious to see if there are different levels of genetic diversity in different estuaries. There are some subtle things that I'm not going to talk about, but basically the idea is if you're living in a place like New Bedford Harbor and everybody who did not have the genes that let you tolerate PCBs, we predict that there should be very low levels of genetic diversity left because you've been through an evolutionary or a genetic bottleneck. Other questions we're interested in is, are some alleles more common than others in polluted habitats? We're starting with some of the sequences that are known to code for that biochemical cascade that I just showed you. And then I'm curious to see if population level evidence is consistent with lab and sequencing studies that show that this variation in some of those alleles is important in the real natural world. No, I think the evidence is pretty good, and I think we'll certainly find that, but it would be nice to find how those things are distributed across a whole landscape. Our study plan, we're collecting these fish from a mix of clean and polluted habitats in New England. I'll show you a map in just a second. We're using two DNA tools. One is a DNA fingerprinting technique called SNPs. I'll talk briefly about that. The other thing is we're doing direct sequencing of those loci that are involved in pollution tolerance. That is what Camilla is mostly focusing on this summer. Here are collecting sites. Thanks to the work of the EPA, we have a good idea about pollution levels at each of these places. Red is higher, yellow and white and green are lower. I'm sorry, white actually means we have no data, but we are kind of assuming because they're close to the low level places, they're probably low. We've collected from all of these sites, and we've got 50 fish who have been turned into 50 DNA samples from each of those places. We do have a couple more sites that we want to visit, hopefully this summer, maybe not for both. This site is Little Compton, Rhode Island. We will go there very soon and finish up our collection for this year, we think. This site, I mentioned that there's parallel evolution. Well, this site up here is Cranston, Rhode Island. It's next to a former Ciba-Geigy chemical plant that has since been closed. It's not been designated as a Superfund site, but it is definitely a major polluted area that's being remediated. We would like to collect there. I went there a couple years ago with Sam Lomax, and the problem we ran into is that there's no shoreline access. So you can see Matt and Emma hanging out on the shore. We can just throw our traps in. That's all great. Well, in Cranston, in that neighborhood anyway, it's all private property, and we'd have to ask permission. And I don't know. I'm shy. I don't like knocking on strangers' doors. So the solution to that is we need a boat. And nobody's going to buy me a boat, but our EPA colleagues have boats. And before the pandemic, we were planning that we would just go there in their boat, drop a few traps, and then we're done. The EPA has fairly strict regulations about how their people can be present in the labs and together in boats and vehicles. So that's been delayed a bit. I'm not sure if we'll get to that this summer, but it's on our agenda for the future. But again, I think it's going to be an important future part of the story because we maybe will better understand how this thing evolves. Does it evolve in exactly the same way in every place, especially if you're in the same neighborhood, or do different mutations get you to the same place? And there are parallels of that in different evolutionary stories. So that's on our radar. While I'm here, I do want to once again thank our many sponsors. So the SNEC Foundation, we've had funding from them for the past couple years. We were able to pay Matt's salary last summer, and that was a real lifesaver. That rescued a big part of that summer for us. Emma Harrington, who is seen holding this trap, was sponsored by Surf. She did a lot of the computer work to make the programs run that I need to analyze this data and got us through some of the preliminary analysis. So I really appreciate her efforts during our abbreviated summer last year. Matt this year is sponsored by a Walsh Fellowship from Providence College. And then Sam Lomax was sponsored by Surf. And I'll get to Camilla. I haven't forgotten you. You have your own slide. So here's how we collect. So you saw the trap that Emma was holding. It's got little funnels, and we put bait, usually squid in there. The fish swim in. They can't get out. After about a half hour, these minnows swim in, and they're very easy to collect. We take them out, we anesthetize them. We're, per our permits and our IACOC rules, we're doing mostly non-destructive sampling. So what we do is we anesthetize them. We take a small fin clip, either from their tail fin or their dorsal fin. We freeze that, we bring it back to the lab, and we turn that into DNA. We let the fish recover in clean seawater, and they seem to swim away being pretty happy. So Camilla, here's Camilla working in our lab. The red lights are because we're working in an animal room that's designed for circadian rhythm studies in rats. We do have overhead lights that are clear, but these just look cooler. And here she is running an agarose gel and cutting bands out. She is doing direct sequencing on some of the genes that are implicated in this particular enzyme cascade. She hasn't seen these yet, but these are her results from the other day. Look at her smile. I love this talk. So nice job, Camilla. We got some decent results. About half our samples worked pretty well. So I think Matt and Camilla should feel pretty good about that. We're going to expand that part of the project and build a nice big data set from this. Matt and I this afternoon are going to focus on finishing up the SNP data collection. So SNPs are a DNA fingerprinting technique that relies on these single nucleotide polymorphisms, basically variable bases within a known stretch of DNA. We're examining about 200 SNP loci scattered around the genome. This means we're looking at more of the genome, and linkage will be our friend there. But we're looking at more of the genome, but in much less detail than Camilla is doing with her direct sequencing. Some of these may be associated with phenotypic variation that's important. Others may be neutral. The neutral ones will let us track population history. And we were fortunate because there's a whole genome sequence, and that let us really pick and choose different markers so that we can answer multiple questions in this system. One of the things we took advantage of is the fact that we know how many chromosomes there are and we know how they're laid out. So we've got an average of about 8 of these markers per chromosome. That should be a really helpful tool for us going forward. We use this magical machine that I'm very excited about. I'm not going to bore you with all the details, but I will show you the kind of results we get. So what this machine lets us do is do high throughput, high detailed genotyping at these SNP loci. What you're looking at here is an array of 96 individuals. So these are rows of individuals at 96 different genetic loci. Each dot represents a genotype at an SNP locus. It might be a G or it might be an A. And the color tells us what it is. So red might be a G, blue might, or green might be an A, and then a heterozygote would be in between as a green. This is a false color thing. I mean, it's not, they don't really look this way, but the system picks it up this way. And so what you're looking at is the end product of 9,600 9,600 PCR reactions that let us genotype 96 individuals at 96 loci. We are, as of today, about halfway through the genotyping. We're going to continue over the next month. To get to this stage, now that we've got the DNA, takes us about 2 days. It's a couple days of prep, but it's a lot of information at the end. It's very rewarding. And the nice thing about this is there's not a lot of post-processing or thinking we have to do. Black, if you're curious, is our negative control. The orange and gray ones are individuals who failed for some reason, but the rest, we just take it out of the machine. Hey, this is an A, this is a T, this is a heterozygote. Comes out in a spreadsheet, and we dump that into our analysis programs. Now, this is a preliminary result. Sam Lomax and I did this 2 years ago. This is an analysis done on a very broad scale with a very tiny number of individuals, maybe 8 or 10 from each of these 4 different populations. What we've done with this analysis is it's based on PCA. We are capturing all the genetic variability and condensing it down to 2 variable axes. And what you're seeing are clusters of individuals that represent individual collecting points. Each dot here represents an individual condensed into this artificial 2-dimensional space. The sort of scatter you see is a representation of genetic diversity. And then where these clusters are show how similar different populations are to each other genetically for this particular subset of loci. Now, I don't want to get too carried away with this story right now. One of the most dangerous things that population geneticists can do is get too excited about partial pilot data. There's a real tendency to stop when you have a cool story and not stop until you do. I don't want to do that. So what we'll do is we will collect we're not going to work on a broad scale with this project. This was really a pilot to show the technology works. We will return to our very focused, detailed biogeographic story from Rhode Island and Massachusetts and share that with you, hopefully, at the SURF conference at the end of the summer. I once again want to thank and express my appreciation for my research students, for Diane and Brian for inviting me into the system, and for all our funders. For those of you who are curious, I can zap this to you if you like. I made up a little bibliography of resources I was pulling together as I put this talk together. And with that, I'm happy to wrap it up and let you go or take a few questions.
Thanks, Jeff.
You're welcome.
First round of applause. That was wonderful.
Thank you. It was fun.
I'll open up to questions from our students before I pepper you with a few of my own.
Sure. And and honestly, guys, the answer might be I don't know. I've been doing this for a couple. I know. I'm sorry. Well, you can take Matt and Camilla's money back then.
I wouldn't do that to them.
Yeah, I mean, because I'm like I say, I'm flattered to be involved in this system, and I'm learning a lot. But man, I have a lot more to learn.
Oh, we all do. So, any questions from our group out there?
I have a question.
Sure, Cynthia.
So I'm kind of interested in mushrooms, and I've read a lot of literature on how they're very useful to clean up water pollutants and other types of pollutants. So I was wondering if you knew of any research currently that has been conducted to use mycelium to clean up water contaminants.
I don't. I do have a question for you. I've heard about similar things with plants, right? They suck the heavy metals out of a polluted site.
Yes.
Yeah. What would you do with the mushrooms once that happens?
Yeah,
go ahead.
Okay, this is a bit of a story. I went to school, and behind my school we have a lake.
Yep.
And the lake used to be polluted because before there was a factory there, and it polluted a lot of the water. So
okay, where is this, Cynthia?
This is in Elmwood Reservoir in Providence.
Oh, very cool.
Yeah, so I didn't know we had foxes. It's a whole ecosystem behind our school. So I was working with my AP biology teacher, and he was telling us how the water contamination was lower now. But then I was always interested because there were a lot of mushrooms in the area, and I'm like, is that good? Is that bad? Because when I was younger, I'm like, mushrooms are poisonous. They're bad for you. But then you learn more, and you're like, okay, they're great for the environment.
Yep.
So then I became interested in how they're used to clean up pollutants. And I was talking to my dad before, and he was telling me, because in my country, when you look over one of the bridges, there's a lot of plants in the water. And my dad was telling me that they were bad for the environment because they were contaminating, holding a lot of pollutants in there. And I'm like, okay, there's a lot of connectivity. So then I went to the mushrooms. How do they help with cleaning? Is it extended to the water? And I was reading, and they're saying that it works to remove E. coli from slow moving water, but they haven't been able to reproduce that with fast moving water. So then I'm like, is anyone else doing research on this? And I feel like mushrooms are not being explored as far as we could. And I'm just wondering if anyone out there is doing research, so I can get in touch with them and see what they're doing. Because I don't know how to start research by myself. So
yeah, I mean, that sounds really cool. And I honestly don't, you're sharing this with me for the first time. This is the first I'm hearing of it. That sounds really, really neat. I have a colleague here named Laura Williams. She may be away this week, but she's a microbiologist, and I think those are the people I would talk to if you're, especially with the E. coli. when I see Brian or Diane next, I'm going to try and remember to ask them too. It sounds very cool. I really don't know anything though. That's neat. Thank you for sharing. Yeah, yeah. Laura Williams at PC. And you can email me or drop me, and I can connect you at some point. Now I don't know that she knows anything, but she knows a lot of things.
Okay.
Yeah, I mean that's it. Yeah, very cool. And maybe bioremediation in New Bedford Harbor means just harvesting all the killifish and taking them out of the system. Wow. Okay, cool. Any other questions?
I have a question. So do you consider humans to be the driver behind this like rapid evolution you talk about? Or do you think of it as like natural selection? And like where's the line between the two?
Here's the thing. I'm branching out. So for years I've taught general bio labs at PC. Someone's going on sabbatical, my friend Elizabeth, and I'm going to teach her evolution class. And I'm starting to reread Darwin and remember stuff. I don't think there's too much of a line, honestly. Selection is selection. And when I read Darwin a few years ago, I remember he talked about you have a herd of sheep and you select the sheep that you want to reproduce because it has traits you like. And he calls that, I think he calls it artificial selection or agricultural selection. And I think natural selection's become a word like like fireplace. Like it literally means nature is doing the selection, or artificial selection means we are doing it deliberately. I think this pollution stuff is somewhere in between, but I think really it's all kind of the same thing. I do think that we have to be thoughtful that, first of all, there's only one fish out of many that managed to do this. So I still think there's a cautionary note. It's not like everybody's going to evolve out of our mess. But yeah, so I think it's a good question, but to me, in my mind, it all kind of comes out the same. And since there are lots of examples of rapid evolution in response to natural circumstances and rapid evolution in response to human-made problems, to me, it all comes out the same. But that may be because I'm a population geneticist and I am a terrible natural historian.
So, Jeff, you gave two examples today of fish that seem to handle adverse situations better than others. So the tilapia in the Salton Sea and the killifish in the polluted bay areas, right? Yep. Is there something about those two species in terms of their underlying level of genetic variation to begin with, or their evolutionary history? Like, why those fish?
I don't know. I don't know. We might actually come up with one explanation. So the party line on killifish is they don't move much. And that's true. When you mark them and recapture them, they're usually 100 yards in a lifetime. That's it.
Oh, wow.
However, that doesn't mean, it doesn't take many migrants between habitat patches to really mean you have one big population. A couple migrants per generation will be enough to homogenize all the genetics over time. So killifish, you've seen them. You walk along any estuary, you see them basically following you. It may be that population is so ginormous and connected that they have the resources to do that. And people have studied, I don't want to pretend that I'm a pioneer in this. People have studied this stuff with other markers, but they haven't looked at it at the same sort of fine scale with this marker that we are. So that may be part of the story for the killifish. Another part of the story, just for educational purposes, is Brian has a nice review paper that points out that adapting to one pollutant is easier than adapting to multiple pollutants. So a situation like New Bedford Harbor, killifish got lucky and everybody else didn't. They had what you needed to adapt to both the heavy metals, the various classes of organic pollutants, and that all worked out. Salton Sea, I got no clue, because First of all, the marine fish couldn't tolerate the environment. No, I
Right.
know you know more about marine stuff than me. I'm kind of guessing that because the marine fish couldn't hack it, maybe because, well, the marine environment's stable.
Yeah.
Tilapia live in brackish water, so they already need to osmoregulate a bit. What the flaw in my story with that is they were escaped farm fish. I know farmed tilapia, the tilapia people are terrible at managing genetic diversity.
Right.
You
I
know,
was going to say that. Yeah.
the cow people are awesome at it. The breeders all worry about inbreeding depression and make sure they're outcrossing and all this good stuff. Tilapia people are my biggest fish. I'm going to sell that one and I'm going to then use these little leftovers as brood stock. And there are tons of bottlenecks and weirdness. And there were certainly more of them in the 1960s. So how they're managing to do that, I don't know. A few years ago, one of my PC students looked at the genetic diversity. There does seem to be sort of an ongoing bottleneck in the Salton Sea. So, and that's a lot of hand waving, Jim, but I don't know. I don't know. But I also know from a sort of conservation environmental point of view, it's kind of useless to think that way because the truth is killifish did it. Hardly anyone else did. You know, so
Right.
is that all helpful? I'm not.
Oh
I'm
yeah,
guessing
it's interesting for me anyways, from just maybe just an academic evolutionary thought process of like
yeah.
what was like why the killifish and nobody else, like what there had to be, like
Yep.
there has to be a reason. I believe that, right?
Yep.
That there was, and whether it was circumstantial, that it was they happened to have the right particular right mutation and a particular part of that cascade that allowed them to survive initially, and then it just from there the population is large enough,
Yep.
and then you have enough pressure and you have enough diversity, and there you go, right? But but the tilapia was the confusing one because I would assume that there was almost no genetic diversity in the escaped fish. So
No.
maybe as another example of pure luck, right? They just happened to have whatever it was that they needed in that moment.
And I saw you light up. So I didn't say it to the group, but they're not only there, they're thriving. So here, I'm going to indulge myself for a second with the pictures I didn't show. All right. So Salton Sea, modern ghost town, yada, yada, yada. They are now an environmental resource, which blows me away. So coastal California is all developed. All the estuaries, all the wetlands are now condos. That's important for migrating birds. They don't have a place to go. Well, the southern end of the Salton Sea is now basically equivalent to a marine wetland, except the migrating birds don't eat marine creatures anymore. A lot of them are eating tilapia. So they are thick on the ground. It is not hard to catch them. You dip a net in and you got tons of fry. You do a drag net and you've got more than you can lift. There are a lot of them. And just to round out the story and its weirdness, the youths will not get this reference, but one of the Congress people from this area who really fought, there are a lot of environmental equity issues that I don't have time to talk about, but the congressman for this area was Sonny Bono. They named this wildlife preserve after him. He was a Republican, but he was also a very strong environmentalist and seemed to be very involved before he died in environmental justice issues. Sonny Bono was married to Cher, and they had a variety show in the 1970s. He later on became a congressman. That just makes this whole thing really surreal because you're in a modern ghost town. The big advocate is a retired variety show host who then later sadly died by skiing into a tree. Is that right, Jim? I think that's the story. Anyway, it's just this wild environment, and how they're pulling this off, I don't know. And I would like someone to give me money. So if your EPSCoR friends ever want to give me money to work outside Rhode Island, I've got the samples.
Okay.
And and I got to visit this area. Jim had mentioned I used to work for USGS in California, and that's where I got these samples. So yeah, that's the story.
Wow, that's amazing.
Yeah, it's cool. All right.
All right. Any last minute questions or comments, or? we are up against it. We are over 1 o'clock. So
Yeah, I didn't mean to go on quite so
no,
long.
that's what happens. That's what's great about these conversations, I think.
I really, yeah, and I do want to say I really appreciate Jim doing this. This is such a nice compromise between having to trek to a different school to hear these hopefully interesting lectures, but we don't have time for that.
Yeah.
So
No,
I think
agreed.
this was a nice thing that we're doing. All right, thank you, everyone.
Thanks, everybody.
Jim.
Hey.
All right.